The 14th century medieval zombie apocalypse brought upon untold deaths to the living. The Mongol army employed a military tactic that catapulted dead bodies over the walls of their enemies in hopes of killing the inhabitants with the rotten stench of the dead. Unfortunately, unbeknownst to them, the cadavers they were using were infected by the Black Plague. For this reason, many historians credit this form of biological warfare as a driver that spread the infectious disease from modern Crimea to the Mediterranean basin. Let's explore the science behind this fascinating history, how cadavers spread infectious diseases to the living. First, let's explore what infectious disease really is. Well, infectious diseases are caused by germs consisting of a range of viruses, bacteria, fungi, and parasites. This presentation will focus on viruses and bacteria. Viruses sneak into the body by camouflaging themselves from the immune system with a capsid envelope. After entering the human body, the proteins on the capsid interact with receptors on the cell membrane to trigger a signaling transduction pathway that allows the virus to enter the host cell. Once inside, the viral RNA hijacks the cellular machinery to replicate, transcription, and translation. Unlike viruses, which depend on a host cell to reproduce, bacteria are self-sufficient, replicating through binary fission. It is estimated that 5% of bacteria are pathogenic, yet they have caused untold suffering and death. Viruses and bacteria appear to be intelligent, as they are continually adapting to the ever-changing environment and exploiting mechanisms to enter the host. However, the immune system is a formidable opponent. Viruses are typically harder to fight off, as they have already infected the cells. The immune system can be broken down into the innate and the adaptive. People are born with innate immunity, which prevents harmful pathogens from entering the body by recognizing general patterns. Innate cells activate adaptive immunity, in which B and T cell receptors recognize antigens. This process aids in acquiring immunity against the pathogen, allowing the immune system to recognize and destroy the foreign agents. The B cells produce antibodies to block pathogens from entering the cell, whereas the T cells destroy infected cells. This raises an interesting question. How can an infectious disease that is reliant on reproduction spread through cadavers? To answer this question, we will examine the following two cases: Ebola as a representative of viral infections, and the Black Plague for bacterial infection. In a 2015 study on the postmortem stability of Ebola virus, the study used Ebola virus-infected Masaquis to confirm the virus as infectious after death for as long as 7 days, with its RNA being detectable for 10 weeks. Rightfully so, the CDC recommends cremation in order to destroy the cells and prevent any possibility of spreading infection. High temperatures alter the structure of proteins, causing them to become denatured, and temperatures above 60 degrees Celsius are high enough to break apart the proteins necessary for reproduction, thus effectively killing most viruses. Traditional burial preparations required washing the corpse, increasing the transmission risk. For example, a 2016 CDC report found a funeral in Sierra Leone responsible for a cluster of Ebola, with 75% of those infected reporting have come in contact with the corpse. Similar to the Ebola example, there are many bacterial infections such as the Black Plague that are also known to spread through contact. For example, a number of studies involving animal carcasses found that the Black Plague can continue to be infectious 24 hours after death. The Mongols' catapulting of the dead may have contributed to the spread of the Black Plague. However, with proper burial practices, the risk of spreading an infectious disease can be significantly minimized, preventing a zombie outbreak that seeks to turn the living into the dead.